You're gonna see it follow along here with you. So if anything goes wrong, now I can fix it in the moment. Oh, I think there's feedback.
Yeah, but can you make it echo?
I think maybe now. Is it down here? Down here.
That's so odd.
I have it set to be muted too. Hello? Is it still doing it? It is. I don't even know where it's coming from, to be honest. Mute, mute. Now?
I think so.
I think so.
Yep.
Maybe? Is it annoying? Where's it coming from? Down here.
Yeah, I think so.
I think it's this box.
I don't know if it's correct that one.
But you can stand like right here. And
I know, right. It's okay.
Thanks everybody for coming to McKenzie Johnson with us today. Dr. Chinsla. And the PhD candidates. Thank you for defending your proposal. Here. Do you have any questions as a student? McKenzie's a great resource. I've seen her, she is your graduate student. So good source of and I will let Kevin have the rest of the time.
Okay, thank you for the introduction. My name is McKenzie. I am a new PhD candidate. I work on the Chin Lab specifically. Today I'm going to talk to you about mechanobiology of compression-induced malignancy and glioma. Y'all hear me well, even in the back? Okay, good. So I mentioned glioblastoma. Glioblastoma is a stage 4 glioma, and it's the most common primary tumor in the central nervous system. It has an abysmal life expectancy of just 14 months. And this is despite standard treatment, including surgical resection, aggressive chemotherapy, and radiotherapies. One of the schools of thought as to why this tumor progresses regardless of aggressive therapeutic intervention is the ability of this tumor type to shift between different phenotypes. Specifically talking about the different phenotypes in GBM, more specifically between the mesenchymal and preneural phenotypes. The preneural phenotype is a less aggressive phenotype, whereas the mesenchymal phenotype is a highly aggressive and invasive phenotype, potentially contributing to recurrent tumors in the primary tumor space, as well as the development of secondary tumors across the brain itself. PMT is a phenotype that promotes GBM aggressiveness. Specifically, we see it, as I mentioned before, in this preneural to mesenchymal transition. And it can be classified a couple different ways. Specifically, we see that STAT3 activation is an early indicator of this mesenchymal shift, as well as cadherin switching, switching from an expression of E-cadherin to that or P or N-cadherin, which increases the migration of these cells. We also see an upregulation in mesenchymal markers, specifically in GBM, mesenchymal GBM, we see CD44, vimin, and ZEB1 all increased, which all contribute to a more malignant phenotype of these cells. This process has been fairly studied from a biomolecular standpoint. We know a lot of biomolecular cues that contribute to this pathway. However, recent literature has suggested that biophysical cues in the tumor microenvironment can actually influence this pathway from a mesenchymal to preneural shift or preneural to mesenchymal shift, independently of biomolecular cues. So even if we have a therapeutic that targets these biomolecular pathways, this biophysical pathway can still promote this transition and promote a mesenchymal subtype in these tumors, regardless of that therapeutic intervention. And this may contribute to the low efficacy that we see in current treatments. So I mentioned the tumor microenvironment. There are a lot of physical forces that happen in microenvironments all across our body, but we see that these physical forces are all pathophysiologics that are all altered in the tumor microenvironment. Specifically, looking at the application of shear stress, we see alterations in shear stress patterns due to angiogenic growth and leaky vasculature in the tumor microenvironment. And we also see that this altered shear stress pattern promotes migration in mesenchymal cancer phenotypes. We also see alterations in tensile strain with an increase in tensile strain around the perimeter of the primary tumor. We also see that tensile strain can promote invasion in 3D. So just the application of this one biophysical component can promote invasion. Next, we also know that ECM stiffness is increased in most cancer tumor microenvironments. And as well, we also see that ECM stiffness can promote migration and invasion. All of this to say is that the biophysical tumor microenvironment is very prevalent and can act as a regulator of malignancy and evolution. The problem here is that these are all end-stage mechanics. These are all biophysical alterations that we've seen when the tumor has been discovered. There has been little study and now more emerging studies on the emergence of the biophysical aspects of the tumor microenvironment and how they play a role in promoting this mesenchymal phenotype from the beginning, from the development of this tumor as through progression and eventually finding. That's kind of my question here is that what are the forces during emergence that contribute to a more overall malignant and mesenchymal phenotype? We're talking about emergent forces. One of the biggest forces that can impact all of these other different physical aspects of the microenvironment is compressive stress. Compressive stress is present from the development of the primary tumor. Think of a small tumor that's in this tissue, and as it grows, it's going to experience some strain from the surrounding tissue, some resistance, which induces compressive stress intratumorally. This compressive stress will increase as the tumor grows at a pretty rapid rate. We also see that there is no direct measurement of compressive stress in a patient. The closest we've gotten right now is to indirectly measure via stress relaxation after primary tumor excision, which gives us approximate values of the compressive stress in these different tumor types. So we know that there is an existence of compressive stress and that it is increased in these tumors, but we also see that it can have some pretty interesting effects on PMT and malignancy. Specifically, this study looked at breast cancer and applied around 700 pascals of compressive stress over 20 hours and showed that compressive stress alone can lead to an increase in migration, which is a mesenchymal associated cell behavior. We also see in another breast cancer line that the application of compressive stress promotes mesenchymal marker expression. I mentioned cadherin switching in the background a little bit. So we see a reduction in E-cadherin here and an increase in N-cadherin, as well as an increase of VMN, all signs of a mesenchymal shift. When we're talking about compressive stress and the impact it has on cell regulation, we want to dive into exactly how that could happen. And specifically, when a cell is being compressed, its nucleus is also being compressed. Now, the nucleus is one of the biggest organelles that can mechanotransduce different biophysical forces in the cell. So we see that as it's being compressed, it is actively, this is a mechanosensitive pathway specifically. We also see that with the compression, we also see an increase in DNA damage, which has been shown to drive malignancy, specifically with the increase in compressive stress. Through the decrease in cell height, we see an increase in overall DNA damage. Further, we've seen that there are protective mechanisms against DNA damage we've seen from a biomolecular standpoint, but there are also some that have been suggested from a biophysical standpoint. One of those protective mechanisms against DNA damage is heterochromatin formation, which has been linked to some of the mesenchymal transitions in other cancers. And that kind of leads me into my proposal objective. And the overarching goal here is to elucidate the mechanism by which compressive stress promotes malignancy. Initially, I want to do that by developing tools. In order to study this mechanism, we need tools that allow us to study it. And that is through the development of a 2D and 3D system with a biomimetic hydrogel for cell encapsulation. Next, using those tools, I want to observe and quantify changes after exposure to compressive stress and observe whether or not we're seeing this pernal to mesenchymal shift, specifically through changes in the cytoskeleton, DNA damage, mesenchymal markers, and migration. Next, I want to evaluate the mechanism by which compressive stress produces or promotes a mesenchymal shift, and do that through targeting the heterochromatin formation axis to reduce the pro-malignant effect of compressive stress. All of that works in tandem to pinpoint a pathway and potential therapeutic target in which we would be able to disrupt the pathway by which cells can feel compressive stress and reduce the pro-malignant effect of compressive stress. Here are my aims in order to achieve that. The first one is to develop an in vitro device that applies variable compressive stress on cells in a 2D and 3D environment. The second, to determine how compressive stress drives phenotypic transitions through cells, cytoskeletal and nuclear transduction. And the third, to elucidate the mechanisms of compressive stress induced malignancy through nuclear deformation-dependent chromatin condensation. So we'll start with aim one, which is tool development. I mentioned that I wanted a 2D and 3D design. The 2D is because it allows for ease of investigation. Very easy to take this cell monolayer and do various biomolecular investigations, which allow us to really suss out what's going on. But next, we have a 3D in vitro compression device, which offers a more biomimetic look with the inclusion of an HA hydrogel and multi-axial stress. It's more biomimetic to that of what actually happened in patients. So we'll start with the 2D in vitro compression device system. It begins with cells on a 0.4 micrometer pore size 24 millimeter transwell. This transwell is specific because the 0.4 micrometer pore size allows for ion exchange, nutrient exchange between the cells and the media below, but does not allow for migration through the transwell itself, maintaining a healthy and non-hypoxic cell monolayer that is actively being compressed. Next, we have an agarose cushion. This just ensures that the compressive stress is equally felt across the entire top of the cell and not just the tipmost top. So it allows for equal compressive stress application. Then we have a 3D printed nylon 5 cup. This is a bioinert material, so it doesn't interfere through degradation or anything with the cells themselves, and allows us to put various weights on top of them. Next is our weights. We use tungsten carbide discs, and they're formulated, or they're designed so that each disc applies approximately 200 pascals in compressive stress. When developing this system, we wanted to ensure that there was going to be consistent compressive stress across the entire transwell membrane. There weren't going to be any hot spots towards the middle or edges. So in order to ensure that, I made a rendering based on the manufacturer's measurements and material specifications in SolidWorks, and then applied a 35-gram load. As you can see from the transwell membrane, there is a pretty even distribution of the compressive stress across the membrane, ensuring that we're going to get a cell monolayer that's equally compressed. Next, we wanted to validate that the cells in the system were actively feeling compression. So we compressed them for a kilopascal for 72 hours. And we saw that there was a significant reduction in circularity, but a significant increase in aspect ratio of the nucleus of these cells, indicating to us these cells are actively being compressed. We also saw that there was a decrease in Ki67, which is a proliferative marker, indicating that these cells are actively adapting to being compressed. There are bioregulation changes in the cell itself. It also suggests that compressive stress, especially high compressive stress, may contribute to selective survival, which we may see later in this presentation. So that kind of sums up the first part of aim one, which I designed the 2D in vitro compression device. And now we'll move into the second part, which is the 3D in vitro compression device. One of the big components of the 3D in vitro compression device is the HA gel itself. The HA gel incorporation allows us to have a more biomimetic hydrogel than that of polyacrylamide or matrigel, as it is one of the biggest components of typical brain matrix. Mention HA. HA is hyaluronic acid. It's a glucosamidoglycan and one of the biggest components in ECM of the brain. Specifically, the makeup of HA in the brain and the tumor microenvironment in the brain is controlled by HA synthase and hyaluronidase, which can either synthesize or break down HA in the tumor microenvironment. It's also important to know that HA molecular weight is pertinent when talking about building a biomimetic hydrogel. Specifically, we see that low HA or low molecular weight HA is more involved in remodeling, whereas high molecular weight HA is more seen in maintenance. In order to build this biomimetic ECM, we could use the typical tenacin and link protein binding as seen in typical brain ECM. However, that doesn't allow us a lot of control to control the stiffness specifically. So instead of using those, we used a chemical called DTT, which binds to the HA and allows us to control the stiffness of this matrix specifically. We can do that through varying the HA weight percent, as well as the thiol ratio, the ratio of DTT. This is done through a Michaels type addition in which the HA is methacrylated to offer a binding site for DTT to bind on both ends, connecting the two different HA strands. Specifically, increasing the weight percent or increasing the thiol ratio, the ratio of DTT allows for a stiffer hydrogel, whereas decreasing them allows for a non-stiff hydrogel. This allows us unique control in order to make a lesser stiffness hydrogel, which is more biomimetic of an emergent tumor, or a higher stiffness hydrogel, which is more biomimetic of an end-stage tumor environment. We also added RGD, which is a peptide that just promotes cell adhesion to this hydrogel. Specifically, preliminary results comparing against polyacrylamide showed that with the incorporation of soluble HA, we see an increase in cell speed in both the low molecular weight and high molecular weight HA. But when placing cells on the HA hydrogel, we see the most significant increase in cell speed, indicating to us that while the presence of HA is extremely important in creating a more biomimetic environment, having the HA in a mechanically bound environment is more important than just having it insoluble. That being said, the design came through like this, where we have a cell spheroid that would be embedded in a 3D hydrogel with a controlled stiffness or set stiffness that'd be placed in a 3D printed ring. And then the same kind of design as the 2D, there would be an agarose cushion, 3D printed cup, and weights atop. And this allows us for multi-axial stress instead of the uniaxial stress seen in the 2D device. Preliminary study or testing showed that there was a significant decrease in circularity in the tumor spheroids that were embedded in this system, as well as an increase in YAP-TAZ fluorescent density, which is a sign that the cells are not only feeling this compressive stress, but they're actually mechanotransducing this compressive stress in the 3D environment. So we showed that we have these two different tools, and they allow us two different ways to evaluate the role of compressive stress in the tumor microenvironment in vitro, both in 2D and in 3D. Next, I want to focus more on aim 2, and that's using these tools to determine how compressive stress drives phenotypic transitions through cytoskeletal and nuclear transduction. I mentioned earlier that nuclear deformation is key to sensing compressive stress. As this nuclear is swished, it activates several mechanisms by which this compressive stress is mechanotransduced. We've also seen that with compressive stress application, nuclear cytoskeletal proteins, specifically lamin AC, are altered with nuclear compression, indicating a change in phosphorylation and potentially lamin structure in response to the added compressive stress to the nucleus and the deformation of the nucleus specifically. We've also seen that vimentin can act as a protective mechanism against nuclear blooding and rupture, with the loss of vimentin showing a significant increase in leakage and destruction, whereas the wild type, while maintaining vimentin, does not have this. And the vimentin plays a role in protecting or caging the nucleus to prevent blooding, which may contribute to apoptosis. We've also seen that overexpression in vimentin promotes slug, which is a mesenchymal transcription factor, indicating that there may be some relationship between vimentin caging and increasing mesenchymal transcription factors. We've also seen that compression induces migration in pancreatic cell lines, showing that there is biomolecular changes and functional changes to the application of compressive stress, which kind of leads me to my question here in this aim, and that is, does compressive stress promote PMT in glioblastoma specifically? In order to study this, I use my compression platform as shown in AIM-1. Specifically, we use U251 GBM cells, which is a more perneural bulk line. We compress these cells for 72 hours at low, which is approximately 400 pascals of compressive stress, and high, approximately 800 pascals of compressive stress. In order to elucidate different changes, we looked at the structural changes of vimentin and lamin to determine the impact of compressive stress on cytoskeletal and nuclear cytoskeletal proteins. We also wanted to look at the phenotypic changes to compressive stress, and we did that through analyzing the protein expression of different mesenchymal markers. We also wanted to look at the functional behaviors of these cells, so we evaluated changes in both 2D motility and 3D confined migration. Last, we wanted to determine compression-induced alterations to gene expression, which is through RNA sequencing after both 2D and 3D compression. So I talked about alterations of both vimentin and lamin. Initially, we looked at the relative lamin expression and saw that with the application of low compressive stress, we see a significant increase in lamin expression, whereas we don't see this same trend reserved in a high compressed condition, which is really interesting. So we want to take a closer look at this, and we did some immunofluorescent imaging of lamin and vimentin, and we saw a unique kind of characteristic here. With the low compressive stress application, we see that there's this lamin ring formation. You can see in the middle bottom picture here, whereas the lamin is localized to the nuclear membrane itself, whereas lamin was kind of dispersed across the nucleus in the high compressed and the non-treated condition. We also see the formation of long spindling fibrous vimentin strands in the low compressed condition compared to the high and non-treated condition, indicating that the vimentin is forming the cage or the network as suggested in order to protect the nucleus against blooding or DNA damage. So next we wanted to look and see if there was DNA damage specifically. And we saw something really interesting here, that in the low compressed condition, there was an increase in DNA damage, but non-significant from the non-treated itself. But there was a significant increase in DNA damage in the high compressed condition. And this combined with the lamin ring around the nucleus, as well as the vimentin cage, suggests that those two cytoskeletal proteins and the alterations in their structure may play a role in protecting against excessive DNA damage. Next, we wanted to look at two different mesenchymal markers to see if they were upregulated in any way in these cells, and they were in fact overexpressed. In STAT3 and ZEB1, there was an increase in relative expression in the low compressed condition, but not the high compressed condition. This combined with the lamin data kind of suggested to us that compressive stress may act in a dose-dependent manner, meaning that the cells who have not experienced compressive stress may not react well to a high compressive stress off the bat, but it may be kind of drawing back to the emerging tumor phenomenon where there is low compressive stress at first and it increases so that the cells survive this low compressive stress and make these cytoskeletal alterations and make these biomolecular changes within the cell are able to survive higher amounts of compressive stress. We see this trend observed across 2D motility, with cell speed increasing in the low compressed condition, but not the high compressed condition. We see an increase in persistence that is non-significant, but there was no chemokine added, so there is no expectation for persistence to be different across the board. So we see this trend reserved, and we wanted to see if it would be reserved in a more biomimetic setting. And if you've seen Landon's seminar talk, you've kind of heard of this before, but we use microchannels. So specifically, I use 5 and 50 micrometer wide microchannels. These are PDMS microchannels that we coat with fibronectin to allow cells to travel in between them. Interestingly enough, we see the same trend reserved with the low compressed condition. The cell speed increases in both the 5 and 50 micrometer wide channels. The 5 micrometer channel is more reminiscent of a tumor microenvironment in which there is really low density and small pore size, whereas the 50 is more biomimetic to that of the surrounding environment of stromal tissue, in which there is a larger pore size. We see a significant increase in low compressed condition, more significantly in the 5 compared to the non-treated, indicating to us that while there is an overall increase in speed and compressive stress, that it's more significant in the pore size that's more uh, reminiscent of the tumor microenvironment itself, suggesting that compressive stress may promote invasion out of the tumor itself into the surrounding tumor. That being said, we've gone through the changes in laminin vimentin as well as DNA damage. We've seen an increase in these PMT markers with the application of low compressive stress. We've also seen changes in motility and confined migration in response to the application of low compressive stress. The next thing to do is to look at RNA sequencing to look at the gene regulations in response to compressive stress. And to do that, we would use the same experimental design and then take the lysate and use RNA sequencing to look at the differences in genetic expression, what's being upregulated and what's not in response to compressive stress. We also want to use this in the 3D system to see if there are any differential effects of using the 3D rather than 2D. And that kind of leads me to the summarization of aim 2. We showed that there are alterations in overall laminin expression as well as structural changes between laminin and vimentin with the application of low compressive stress. We've shown that low compressive stress promotes mesenchymal markers and that these alterations in cytoskeletal structure may contribute to the protection against DNA damage. We also showed that there was an increase in migration in both 2D motility and confined migration, indicating that there's a functional change. These cells are behaving differently. And lastly, we anticipate that genetic expression contribute to increase in migration and therapy resistance in response to low compressive stress, but not high, which is in line with what we've seen so far. And that kind of takes me through to aim 3. And in this aim, we want to elucidate the mechanism of compression-induced malignancy through nuclear deformation-dependent chromatin condensation. One of the new fields of study in bioengineering, specifically in mechanobiology, is this idea of epigenetics. That's the idea that we can put these cells in an environment and they create this mechanical memory. They learn from these environments. So say you put a cell on a stiff substrate and then you put it on a soft substrate, it's going to have some changes from a cell that's only been on a soft substrate. So creating these epigenetic changes. We've seen that mechanical stimuli can alter genetic expression specifically, and it does that through genes transcription controlled by acetylation, deacetylation, and methylation, demethylation of histones specifically. This is a dynamic process which regulates the availability to transcribe genes. Specifically, we have euchromatin and heterochromatin. Euchromatin is a less dense, non-methylated version of the histone in which DNA is actively able to be transcribed, whereas heterochromatin is tightly wound DNA around the histone, which does not allow for the DNA to be transcribed specifically. I mentioned in the background that heterochromatin actually contributes to protection against DNA damage. But we've also seen that excessive heterochromatin formation can lead to cell cycle arrest and apoptosis if all of the DNA that these cells need to transcribe in order to function appropriately is being locked up by the histones being methylated, then that makes it really difficult for those cells to survive. Specifically in the context of compression or specifically confined migration, we've seen that the application of a small compression event, as I would call it, or a confinement event, increases heterochromatin marks, specifically at different residues. We've seen that different residues and different methylation of different residues can contribute to changes in cell behavior. Specifically with confined migration, those showed an increase in histone 3 lysine 27 trimethylation. They showed an overall increase in heterochromatin. They also showed that the increase in heterochromatin contribute to confined migration efficiency. I mentioned that residue specifically because it has some role in the mesenchymal transition specifically. Histone 3 lysine 27 is methylated by a polycomb recessive unit, as shown here. One of the main components of this unit is EZH2, which is enhancer zesty of homologous 2, and is a transferase that's shown to promote EMT in ovarian cancer. This methyltransferase is specific to histone 3 lysine 27 trimethylation. We've also seen the overexpression of EZH2 is shown to increase invasive capacity, suggesting that this may play a role in promoting mesenchymal transition. And further, that E-cadherin depletion is associated with the downstream effect of EZH2 nuclear localization, which is histone 3 lysine 27 trimethylation, suggesting to us that this may be a possible avenue in which there is some mechanical responsibility of EZH2 localization, which then can contribute to a mesenchymal transition. In order to assess that, we first wanted to look at whether or not we were seeing an increase in histone 3 lysine 27 trimethylation, and we were, but more dramatically in the high compressed condition. But as I mentioned, it's kind of a dynamic process, and that the over trimethylation of histone 3 lysine 27 or over heterochromatin formation can contribute to cell cycle arrest and apoptosis. However, we did see an increase in the low compressed condition compared to the non-treated, suggesting that while it may not be as dramatic as the high compressed condition, we are still seeing some of this specific heterochromatin formation in the low compressed condition. In order to assess if this was a significant pathway in which these cells were mechanotransducing compressive stress, we applied an inhibitor, GSK 343, which is EZH2 specific. With the application of this inhibitor, we see that the pro-malignant effect or pro-migratory effect of low compressive stress is in fact rescued. And with the inhibitor, the cell speed is similar to that of the non-treated or high compressed condition, indicating this pathway is extremely involved in promoting this change in cell behavior and change in biomolecular regulation within the cell. That kind of leads into the proposed mechanism by which we think this happens. The cell is being squished, and there's some mechanical event that allows EZH2 to be translocated to the nucleus, which increases nuclear EZH2, which then increases the ability to form these PCR2 units, which then allows for an increase in histone 3 lysine 27 trimethylation, which will then downstream increase the more mesenchymal phenotype in these cells. We want to evaluate this mechanism through looking at the translocation of EZH2 and methylation activity to see if we are seeing more nuclear EZH2 and if we are seeing an increase in methylation. Here's just kind of the graphic of that again. We also expect to see a decrease in E-cadherin with the increase of histone 3 lysine 27 trimethylation, which will then suggest that there is a more mesenchymal phenotype. We also want to use EZH2 knockdowns to evaluate EZH2 dependent translocation and the behaviors of these cells that have the knockdown compared to the wild type. And then we want to evaluate the genetic silencing through chromatin immunoprecipitation. As I mentioned, when the genes are tightly bound on these histones, they are not able to be transcribed. So we'd use chromatin immunoprecipitation to look at the genes that are actively being silenced with the increase in histone 3 lysine 27 trimethylation. We do this through a similar experimental design as AIM2 with the application of low and high compressive stress, specifically looking at protein expression via Western blots, analyzing cytoskeletal changes via immunofluorescent imaging, as well as analyzing contributions to EZH2 nuclearization through inhibition at various steps, and then analyzing therapeutic targets through inhibition and knockdowns or knockouts. As I mentioned, we also want to do ChIP-seq, which is chromatin immunoprecipitation sequencing. This allows us to target specifically histone 3 lysine 27 and the DNA bound around those histones. We can then separate the DNA from the histone and then sequence it to see which genes are specifically being silenced through this trimethylation event in direct response to low compressive stress and high compressive stress. And to kind of summarize all of this, AIM1, I went through and developed in vitro devices that allow us to apply variable compressive stress on cells in 2D and 3D. Using those tools in AIM2, I've determined that compressive stress drives phenotypic transitions through cytoskeletal and nuclear transduction and promotes a mesenchymal phenotype. And then lastly, AIM3, I want to elucidate the mechanisms by which compressive stress induces malignancy, specifically through nuclear deformation, chromatin condensation. That is my main project and what I've been mainly working on for my thesis, but I'm also involved in a couple of different projects that I think are very interesting and I wanted to share. Specifically, the first one is looking at stromal derived factor, or SDF, and promoting PMT and cell locate or cell migration to the subventricular zone of the brain. We have some interesting data there suggesting that this is a potential avenue in which we can increase patient life, or quality of life, as well as time of life after diagnosis. Specifically, I'm looking at using chemokine SDF and its effect on the expression of PMT markers, as well as receptor changes and changes in 2D and 3D invasion. Another project I'm kind of working on hand in hand with one of our master's students currently is the insertion of allogeneic force probes into tumor spheres in that HA gel that I mentioned earlier, in order to directly quantify the changes and forces intratumorally and in the tumor microenvironment during progression and invasion. This kind of works by having a force probe that is compressible and using imaging and some finite element analysis to analyze the forces based on the depression of these beads specifically. And then I've also done some work doing AFM data analysis and SCM on changes in the mechanical ultrastructure and ECM signatures in glioblastoma, specifically between the core of ECM or the core of glioblastoma tumors and the rim of glioblastoma tumors. We actually have a paper published out and should have another one coming soon, looking at the effects and how we can use what we find out about these tumors in order to better predict prognosis and therapeutic treatments. I've also been active in publishing and going to conferences to present my work. I go to several a year and published my first author paper this year. So check that out if you want to learn more about compression in the tumor microenvironment. And with that, I would like to thank my lab and our funding sources and you all for being here listening. And I'll take questions.
Questions for McKenzie?
So I mentioned that there is indirect measurement through stress relaxation after tumor excision. It's a paper that went through and tried to approximate values in which compressive stress is in the tumor. They approximated glioblastoma to be around 1,000. So we chose that value initially and then wanted to look at more emergent values, and we started with low compressive stress, which is 400. Yes, kind of.
Results it does low and high compression. Have you thought about looking at the time factor? Does low compression for a longer period of time and similar effects?
Yeah, so initially I did do 24, 48, and 72 hours of compressive stress. We saw the most significant change in cell behavior at 72 hours. And in order to really quantify that, we wanted to give it prolonged compression, which is why we went with 72 hours. But I do think there could be interesting results at a lower time point. I just don't think that it fits into the prolonged compression category, if that makes sense.
Based on your research, honestly,
That's a great question. Currently, we're looking at EZH2, but EZH2 has many other responsibilities within the cell specifically. I think targeting that pathway somehow may be a challenge, but should be really interesting if we can find the way that EZH2 is localized into the nucleus. We can try and figure out a way to stop that nuclear localization, either through binding EZH2 and keeping it cytosol only, or some kind of other avenue.
Is glioblastoma the only cancer that this activity happen in other?
Yeah, so there has been some studies of breast cancer, pancreatic cancer, colon cancer, a lot of the other solid tumors, in which compressive stress is expected to be a big key player in a mesenchymal transition. Those would be more of an EMT. However, those are distinctly different in terms of tumor microenvironment than glioblastoma. So we're trying to see if this is still a phenomenon. other questions? Any online questions? I haven't checked.
Thank you very much, McKenzie.
Thank you.
I'm in on the floor. For 601 students, while I fix this problem. You for the invested and these been, hundred been sent out to the. Office. All that before next. I've got the zones. Yeah. It's long. I'm just like. Yeah, it's just. Yeah, oh. I'm sure I got no. Right, so I'm glad that we go. Yeah, that. Yeah, so. Yeah, I feel it. Downtown. Actually, I don't have to say. Yeah. You know, it sounds like that. This is awesome. I can say I'm not. Something's wrong. Absolutely. Training is off running for that project. Stuff for basically, we're just gonna go shopping. So I'm like. It's like, if you look on a slide, say, she'll say that, doesn't say. I agree, but this is more like questions. Okay. Okay. Number of like hiking Minnesota. No, sorry. You make a research of the. Yeah, there's no classroom in it, so there, most students never study. Yes. Yeah. I'm fine. I did some more stuff. Yeah. Doing some stuff about like my circulation.
Well, I was finding something else.
Yeah.
brainstorm.
I will, you said no.
It's registered dogs. We could. Tomato for. Actually, she did it. Did you break your? Higher.
Really?
I do it. Are you, I don't know. Took over it, so that's what I'm telling myself.
Starting. It's full on that. They're all. I think the big.
That's. So you're in French time, you're in stage three. Like all of, after fall break. Yeah. You don't know.
Or
Not
maybe
sure
it's
about
there.
it, speaking of stuff, but. Doesn't matter if you have ideas, but.